Добрый вечер, дорогие участники встреч вне границ. Меня зовут Шихер Анна, я заместитель руководителя факультета психологии Высшей школы «Среда, обучения». Я рада приветствовать вас на наших уже регулярных встречах вне границ, на которых мы знакомимся с новыми актуальными направлениями в психологии, с теми исследованиями, которые сейчас носят как научный, так и прикладной характер, с возможностями актуального использования опыта коллег зарубежных, возможно, в практике научной или теоретической. Рада приветствовать вас на этом мероприятии, которое вновь показывает, что с помощью дистанционного формата мы можем преодолеть любые границы, и действительно нет никаких барьеров в контексте профессионального развития. Сегодня я рада приветствовать нашего прекрасного спикера Джонни Уоллеса, профессора Калифорнийского университета Беркли, с темой очень важной, очень актуальной, а именно нейропсихологии и ее связи с посттравматическим синдромом, расстройством, посттравматическим ПТСР, потому что тема действительно очень актуальная. Я знаю в том числе, что среди многих участников и студентов факультета психологии, абитуриентов часто встречаются запросы на данную тематику, потому что это действительно тема, с которой мы можем столкнуться в обычной жизни, с которой сталкиваются клиенты, которые приходят к психологу. И, конечно, очень важно иметь хотя бы базовое представление о том, как это состояние проявляется в контексте нервной системы, нейропсихологической стороны психики человека. Поэтому я рада приветствовать нашего сегодняшнего спикера. На протяжении всей встречи не стесняйтесь, пишите свои вопросы в чат. На протяжении всего процесса я буду с вами находиться на площадке. И после того, как Джонни расскажет тот материал, который подготовила для нашей с вами сегодняшней встречи, мы откроем сессию вопросов, в рамках которых те вопросы, которые непосредственно относятся к теме, мы постараемся задать напрямую для того, чтобы у вас была возможность узнать самую актуальную информацию из первых рук человека, который напрямую занимается этой темой, непростой, но очень актуальной. Поэтому пишите в чат свои вопросы, они обязательно будут собраны. Если у вас на протяжении лекции возникают какие-то вопросы, тоже пишите. Старайтесь только, если вы в середине делать, указывать контекст. И я также попрошу писать на русском, так как мы используем синхронный перевод, и для удобства это будет оптимально, если вы будете писать вопросы на русском языке. Спасибо вам большое и прекрасной встречи!
Okay, thank you very much. So today I'm going to be talking about one of the most modern neuroscientific techniques that we have available to us, which is a brain-machine interface. And I'm going to be explaining what a brain-machine interface is and how we can use it to study cognitive processes. And in particular, the cognitive process that I'm most interested in is value-based decision-making. So how we make everyday decisions in our lives. So the area of the brain that I'm particularly interested in and going to be focused on is the frontal lobe. And actually within the frontal lobe, it's going to be the underneath surface of the frontal lobe, which we call the orbitofrontal cortex. So this is going to be the outline. I'm going to introduce you to the orbitofrontal cortex and brain-machine interfaces. Then we'll talk about a little bit about how we can use these interfaces to read out information from the brain and also potentially write in new information. I'm then going to introduce another area of the brain that we've recently realized also has an important contribution, which is the hippocampus. And we'll look at what contributions the hippocampus makes to decision making. Okay, so let's begin then with looking at the orbitofrontal cortex. So this is the underneath surface of the frontal lobe. Well, the underneath surface of the brain. So here we have the cerebellum at the back. This is the temporal lobe. And then up here is the orbitofrontal cortex. So this is the underneath surface of the frontal lobe. And we know that this area is particularly important for decision making. And in fact, it's one of the cleanest examples we have of the localization of a high-level cognitive function. So damage to the orbitofrontal cortex in humans produces very specific impairments in the ability of the patient to make decisions. It doesn't affect any other cognitive processes, and we only see that pattern of deficits following damage to the orbitofrontal cortex. Damage to any other cortical region doesn't produce those problems. We also know that the orbitofrontal cortex is dysfunctional in many neuropsychiatric disorders that involve an element of impaired decision making. So that includes things like mood disorders, addiction, eating disorders, obesity, and OCD. And in fact, in this talk, I'm going to be really focused not on PTSD, but on addiction. But those things often go hand in hand. Those things are often comorbid with one another. So I hope it'll still be useful for you. Okay, so let me give you a flavor for what an orbitofrontal patient is like in the clinic. So this is a case study. This is patient Elliot. Sorry, one moment. I need to turn my phone on. Sorry, I should have done that before the talk. Okay, so this is patient Elliot. He had a tumor in the ventral medial part of his frontal lobe. And when that tumor was removed, he suffered damage to the orbitofrontal cortex. And you can see that damage here, down here, and a little bit of the lateral surface as well. So Elliot had, this is how he presented in the clinic. There were no sensory motor problems. He had normal language. He had a high IQ, good memory, and he performed normally on many of the standard tests of frontal lobe functions, so things like the Wisconsin card sorting test. He was a successful person. He was in his 30s. He was an accountant in a building firm. He was married. He had a family. And everything had been going very well for him up until this tumor. So you take him into the clinic and he seems completely normal. If I had damage to my orbitofrontal cortex, you wouldn't be able to tell. I could give this talk very easily. But within a few months of this operation, he had quit his job. He'd been at that firm for 17 years. He quit his job, went into partnership with another man who was actually a con man, and ended up losing his life savings to this man. He divorced his wife, and a few months later remarried another woman. She was a sex worker, and that marriage only lasted for a few weeks. He then moved back in with his parents. He found another job that was 100 miles away from his home, and he was fired from that job because he couldn't show up to work on time. So, having had a very successful life, within a few months of this operation, his life was just a complete disaster. And you might, think that that's just coincidence, but we see this pattern over and over again with patients who have damage to this area. They just make terrible decisions following the damage. And you can see this deficit in the lab if you use decision-making tasks. And in fact, people have stripped them away to make them the most simple decisions possible. And so one example is color preference. So if I hold up two different colors and ask you to choose your favorite color, most people can do that with no problem. And although everybody's preferences are different, our preferences show consistency. So if I was to ask you your preferences today, and then ask you your preferences in a month, you'll give me highly correlated answers. And preferences also tend to be transitive. So if I prefer red over green, and I prefer blue over green, blue over red, then I know I'll prefer blue over green. Now, in contrast, orbitofrontal patients, they seem to choose more or less at random. So their preferences, their decisions that they're making, the preferences that they're telling the experimenter, they seem to be just random. Okay. Now, as I mentioned, we also know that the orbitofrontal cortex is involved in a lot of psychiatric disorders. And here's a couple of examples. So this is the change in cortical volume that's seen in depressed patients. So you can see a reduction in the cortical volume down here in the orbitofrontal cortex. And you also see some reduction in the temporal lobe and the cingulate cortex, but it's most noticeable in the orbitofrontal cortex. These bottom slides here show coronal sections through the brain, and it's showing where the serotonin transporter is located on those coronal sections. And we know that serotonin is very important for depression. A lot of our antidepressant medications target this receptor, and you can see that it's really concentrated down here in the orbitofrontal cortex and on the medial surface as well. So one of the procedures that has been used to try to target orbitofrontal cortex and to try to manipulate it is a technique called deep brain stimulation. So deep brain stimulation is a technique where we implant electrodes into a patient's brain, and those electrodes can then stimulate that part of the brain. The electrodes are connected to two electronic stimulators and battery packs, which are implanted in the patient's chest cavity. And everything is connected via wires which run underneath the patient's skin. And if you've not seen this before, it's very effective for treating Parkinson's disease. So I'm just going to play you a couple of videos that show a Parkinson's patient. So before the video even starts, you can notice that this patient here, he has a somewhat stooped posture, and that's indicative of Parkinson's disease. He's going to have a tremor of his hand, and the doctor is asking him to walk to the door, turn around and walk back again, and you're going to see that he has a lot of difficulty with that, and he also has what we call a shuffling gait. Okay, so you can see that shuffling gait. If you look at his left hand now, you can see it tremoring. He has a lot of difficulty turning around. That left hand is still tremor. He also doesn't show any emotional expression, so we call that a blunted or frozen affect. So a very classical Parkinson's patient. Okay, this next slide is the same patient. He's now been implanted with the electrodes, and the stimulator is on. The electrodes are actually in a part of the brain called the basal ganglia. So you can see he has no problems walking, no tremor, and he also has a big smile on his face, so he doesn't have blunted affects anymore. So deep brain stimulation has been a very effective treatment for some neurological disorders, and not surprisingly, people have tried deep brain stimulation in the orbitofrontal cortex to see if it provides alleviation for things like OCD or depression. And the problem is that so far, it's really not had very much, we've really not had very much success. So what is the difference then between these two structures? So the caudate nucleus is part of the basal ganglia, the orbitofrontal cortex is part of the cortex. And the reason we think is because the cortex works very differently from the basal ganglia. The pattern of neural firing in orbitofrontal cortex is very precise. And when we stick a stimulating electrode, all we're doing is providing a high frequency current into that area of the brain. And probably for a cortical region, that high frequency current is just noise. It just disrupts the region rather than helping it to function properly. So in order to get around that problem, in order to interact with the orbitofrontal cortex in a meaningful way, what we're trying to do is we're trying to use brain-machine interfaces. Now, brain-machine interfaces were developed mainly with respect to the motor field. So this is how a brain-machine interface works. Shown here is a monkey. Most of the research that we do is in monkeys. So in this particular example, the monkey has electrodes implanted in his brain. The pattern of neural activity, in fact, the electrodes are implanted into his motor cortex, which is responsible for movement. The pattern of activity is recorded. It's then sent to a computer that decodes that pattern of activity, interprets that pattern of activity using sophisticated algorithms. And those algorithms are then translated into some kind of interaction with the machine. In this case, it's a robotic arm that the monkey's going to use to touch a cursor on a computer screen. So there are about 250,000 individuals in America with spinal cord injury, and one possible treatment for those injuries would be to decode activity in the motor cortex and then use that activity to control machines. So things like a patient's wheelchair or a computer cursor, allowing them to interact with their environment, or as in this case here, robot arms. So I'm going to show you a couple of examples of this, a couple of videos demonstrating these brain-machine interfaces. So the first video is of a monkey. The monkey's actually sat here. His arms are restrained. So his arms are in these two tubes, which prevent him from reaching over here. The experimenter is going to put a treat on the end of this stick. It's actually going to be a piece of marshmallow. And then the monkey has an implant. We're recording activity from its motor cortex. And that neural activity is going to be used to control this robot arm. And so the monkey is going to use his motor cortex to control the robot arm so that he can get the treat into his mouth. So you see that the experimenter puts the treat on the stick, and the monkey is using that robot arm. So he's using the patterns of neural activity in his motor cortex to control the arm. And you can see that he can use it. He can pretty much reach to any area of space. He can then do a precision grip with the robot arm, and he can then transport the arm to his mouth so that he can get the treat. So this has also been done in human patients. And in the next slide, I'm just going to play a short video which describes some of this work by some of my colleagues.
In this paper, 2 people with tetraplegia, and it's 2 people who are unable to move their arms or their legs in any functionally useful way, were able to control a prosthetic or a robotic arm simply by thinking about the movement of their own paralyzed hand. And they did that using the investigational brain-gate neural interface system. So they thought about using their own arm and hand as though they were reaching out themselves with their own limb, and the robotic arm moved much the way their own arm would have moved. One of the long-standing questions, not only in neuroscience, but in neurorehabilitation, is whether the cells in the motor cortex and other parts of the brain, whether they continue to function the same way years after that original injury.
It is possible for people to use their thoughts to control devices, either a computer or a robotic arm. The way that happens is that we implant a tiny sensor, just about the size of a baby aspirin, just into the surface of the brain. And that sensor pick up the electrical impulses from a bunch of neurons. And each of those little neurons are like radio broadcast towers, putting out impulses. And when they get to the outside, a computer translator converts the pattern of pulses into something that is a command.
One of our participants was able to do something that, when all of us saw it for the first time, gave us all pause. She reached out with the robotic arm. She thought about the use of her own hand. She picked up that thermos of coffee, brought it close to her, tilted it towards herself, and sipped the coffee from a straw. And that was the first time in nearly 15 years that she had picked up anything and been able to drink from it solely of her own volition.
There was a moment of true joy, true happiness. I mean, it was beyond the fact that it was an accomplishment, I think an important advance in the entire field of brain-computer interfaces, that it was really a moment where we helped somebody do something that they had wished to do for many years.
Okay, so you can see that we've had some success then in using these brain-computer interfaces, in this case, for giving someone some degree of motor control that they otherwise have never had, or currently didn't have. Okay, so the research objective that's going on in my lab is to try to adopt these same brain-machine interface approaches to treating neuropsychiatric disorders. So the idea is that, can we modify some of the maladaptive behaviors that underlie neuropsychiatric conditions, so things like addiction, for instance, by reading out patterns of neural activity from prefrontal areas like the orbitofrontal cortex, and then writing in more adaptive behaviors through patterned electrical microstimulation. So you can see that we're essentially treating the brain like a computer. The brain is, to all intents and purposes, a very complex computer. And so if you want to modify programs within the brain, you have to be able to interact with it. You have to be able to read out information and write in new information. Now, I mentioned that all of the work that we do is in the monkey, and this slide just shows you the reason for that. So these are the different cytoarchitectonic areas in the frontal lobe of the human, the monkey, and the rat. So a cytoarchitectonic area is basically, we look at the organization and types of cells in an area, and you can create maps in this way. And these are our maps of the frontal lobe. And you can see that the map of orbitofrontal cortex, so this is the medial surface and this is the ventral surface. The map of the orbitofrontal cortex in the human looks very similar to the map in the monkey. But then when we move down to the rodent, we see a very different organization. So within primate evolution, within the frontal lobe, the orbitofrontal cortex is one of the older areas, and it is very well conserved across primates, and that's why we use monkeys as our model system. Okay, so how do we go about reading out decisions from neural activity in the brain? So what I'm going to talk about is all going to be single unit, what we call single unit neurophysiology. That means we're recording electrical events from a single neuron, and we do that by essentially lowering very fine wires into the brain so that they're next to these neurons and they can pick up that electrical activity. And neurons fire action potentials, which are very brief electrical events, a very quick up and down deflection of an electrical potential. It's about 1 millisecond in length. The slang for those action potentials is spiking, a spike. And so what we're doing is recording patterns of spikes from neurons and then trying to interpret what those patterns of spikes actually mean. So we can do this because the brain actually doesn't contain any pain receptors. So you can insert an electrode into the brain and the animal can't feel it. So here, for instance, we have an animal that is being presented with a stimulus on a screen. And depending on where that stimulus is on the screen, it makes the neuron fire either a lot of spikes. So these are these up and down deflections in the electrical potential, or the neuron only will fire a few spikes. This was actually a neuron recorded in the visual system. That's why it's driven by this light on the screen. And we calculate the number of spikes that are being produced every second to give us a frequency, a firing rate. And that's what you're going to see me referring to in a number of my slides. Okay, so this is the kind of experiment that we do in my lab. We basically have the animal learn associations between pictures and different reward outcomes. So you can see we have a set of pictures up here that differ in terms of the probability that the animal will receive a reward. So this picture predicts getting a reward 10% of the time. This picture predicts getting a reward 90% of the time. We have a second set of pictures that differs in the amount of reward that the animal is going to get. The animals actually work for fruit juice. And then down here we have a set of pictures that varies in terms of the number of, the animal has to earn the juice by pressing a lever a certain number of times, and then he'll get the reward. So in each of these cases, as we go from left to right, the value of the picture is increasing, either because reward is becoming more probable, the animal is going to get a bigger reward, or he has to do less work in order to get the reward. And this is what a typical orbitofrontal neuron looks like when we present pairs of these pictures and ask the animal to make a choice. Now, the animals are really good at this task, right? We give them a pair of pictures and we ask them which do they want, and about 98% of the time, they pick the more valuable option. And while they're doing that, their orbitofrontal neurons are firing. And you can see this is basically the period when the pictures come on the screen and the animal is making his decision. And the firing rate of this neuron increases as the value of the picture that the animal is choosing increases. So what we say is that the orbitofrontal neurons are encoding the value of the different options that are available to you. So now you can understand why patients with damage to this area have a problem making decisions. If they're missing the neurons that are telling them that option A is better than option B, they're obviously going to have to make their choice more randomly. The problem with this, though, is that it doesn't really get us to the decision-making process itself. Now, one problem with studying decisions is that they are hidden from our view. They are cognitive processes. And so that makes them very challenging to measure. And that's just shown in this example here. So this guy here is going to get a choice between red and white wine. And this is his thought processes. 3 different times he's presented with this choice. You can see that he prefers white wine, but each time he makes the choice, he's going through a very different sequence of thoughts. And we don't have access to those thoughts. And that makes it difficult for us to measure this process. Well, one possibility is that we can read out this information from the brain via our neural recordings. We can decode the decision in real time. Now, in order to tell you how we do that, I just need to take a little segue, a little tangent, and explain about neuronal noise. So I told you that these neurons are firing these spikes, and the neurons in orbitofrontal cortex are firing spikes according to the value of the option that the person's being presented with. But what I haven't told you about is it's extremely noisy. So here's an example where we presented the same picture to the same neuron. And you can see that the neuron fires, that the action potentials start firing when that picture comes on the screen. But these are 46 different occasions when we presented these pictures, and the exact pattern of spikes is different every single time. And so what we do is we do this over and over again, and then we average to get the average response of the neuron. However, that's not the only way that you can deal with neuronal noise, and the brain-machine interfaces take a very different approach. And this is what's called a decoding approach, as opposed to the encoding approach that I just described. So in a decoding approach, what you do is, instead of trying to average across trials, you record from lots and lots of neurons, and you kind of average across the neurons. So this now is the activity from a single trial, and we're recording from 40 or so neurons, and we project this pattern of neural activity into a high dimensional space. And in fact, this was recorded from a leech, and the leech was engaged in 2 different kinds of behavior, either swimming or crawling. And you can see that the pattern of neural activity is very different for these 2 behaviors. So what that means is you can look at the pattern of neural activity, and you can know whether the animal is either crawling or swimming. Now, of course, that's not very useful, because you can just look at the animal to determine that. But this is potentially a method that we can get at hidden cognitive processes where we can't simply observe the cognitive process that we want to study. So that's what we did in my lab. So very similar to the previous task, we trained the animal that there were these different pictures that varied in terms of the number of the amount of juice that the animal was going to get. We then recorded from multiple orbitofrontal neurons. Now, it's very difficult to do this because the orbitofrontal cortex is so deep in the brain because it's at the bottom of the frontal lobe. So we were typically recording from about 10 to 20 orbitofrontal neurons by essentially inserting 10 to 20 wires deep into the brain to record the neural activity of these neurons. We then record the pattern of spikes that we see, and we trained up an algorithm to decode the value of the picture that we presented to the animal. And the algorithm was able to do this fairly well. It got the correct picture about 50% of the time, or I should say the correct value about 50% of the time. And because there were 4 different values, that was much better than chance. If it was just guessing, chance performance would have been 25%. So we could decode value information in real time from the orbitofrontal cortex. Now, that's not that interesting. We already know what picture we presented to the animal. But what it enabled us to do is to ask the question, what happens when we present the animal with a pair of pictures and he now makes a choice between them? And what we found is that the neural activity in the orbitofrontal cortex would kind of flip-flop back and forth between either option. So what you're looking at here are 6 different trials where the animal was presented with a choice. The y-axis is the probability that the neural activity looks like one of the patterns that the decoder is able to recognize. And we've color-coded them according to whether it was the higher value option which the animal chose or the lower value option that he didn't choose. And you can see that although it's noisy, there are these periods where the decoder is very certain that the animal is essentially thinking about one or other of these options. And sometimes that activity seems to go through these flip-flops where it starts out in one state and then switches to another state. And we were able to show that these dynamics in the neuronal activity had an impact on the animal's choice. So every now and then, even though the animal would choose the most valuable option most of the time, sometimes he would make a mistake and he would choose the less valuable option. And when he did that, you can see that there were more of these state transitions compared to when he didn't make an error. And we also saw that these states could predict how quickly he was going to make his choice. So in both our subjects, if they strongly represented the higher value option, then they were very quick to choose it in their behavior. But if they strongly represented the lower value option, that really slowed down their ability to make the choice. So in other words, the neural activity, the neural patterns that we were reading out from orbitofrontal cortex were significant for the animal's choice behavior. We could use those patterns to determine how quickly and how optimally the animal was going to make its choice. Okay, so that shows us that we're able to read out information. But what about our ability to write in new information? Well, for this, we did again a very similar task where we present the animal with pictures that predict different probabilities of either receiving or not receiving juice. The difference now, though, was that those pictures were going to change, their value was going to change over the course of the session. And the animal's job was to track those changes and to adjust his behavior accordingly. So you can see here, for instance, at this point in the session, picture 1 is very high value, picture 2 is low value, and picture 3 is somewhere in the middle. Whereas over here, picture 1 is of a middle value, picture 2 is high value, and picture 3 is low value. And the solid line indicates the actual probability that that picture will lead to a reward, whereas the dotted line is the probability that the animal will select that picture. And so you can see that the animal is doing a really good job of tracking these values and adjusting its behavior as soon as the value of the picture begins to change. So you can see here, the value of the picture starts going up. The animal is quickly adjusts its behavior so that it's more likely to choose that higher value picture. Now, when we did this task, what we noticed was that a particular oscillation in the brain, in the orbitofrontal cortex, called the Theta Oscillation, would synchronize to events in the trial. So here's when the pictures come on the screen. These are all of our trials stacked horizontally. And the color indicates the phase of the oscillation. And you can see that the color begins to line up across trials, and that's because the oscillation is synchronized to that event. So let me tell you a little bit about brain oscillations. So these are different from the spikes that I told you about. The spikes are coming from the activity of a single neuron, whereas the oscillations are coming from the spikes of hundreds of thousands of neurons, all interacting with one another. And we have a wide variety of oscillations in the brain at different frequencies, and they've been linked to different kinds of behaviors. So this very low frequency delta wave is associated with deep sleep. This alpha rhythm is associated with being awake and relaxed. So the delta rhythm is about 1 Hertz and the alpha rhythm is more like 10 Hertz. The theta rhythm, which is usually between about 4 and 8 Hertz, has been implicated in a wide variety of things, including, being engaged in a task or thinking or moving. And then the higher rhythms, such as the beta rhythm, which is about 30 Hertz, and also the gamma rhythm, which is even higher still. The gamma rhythm is about 80 Hertz. Those have been associated with cognitive processes. So you can see that for our task, for this decision making, we really only see the oscillation, or at least this syncing of the oscillation in the theta rhythm. There's very little synchronization happening either in the beta or the gamma rhythm. And we also only see this synchronization taking place when the animal is actually adjusting its behavior. So there are periods of time where the contingencies are relatively stable and the animal doesn't have to do anything. And then there are 2 periods of time when the contingencies are changing and the animal has to adjust its behavior. And when the contingencies are changing, we see an increase in this theta alignment in both our subjects. Now, those neurons that I was telling you about earlier, the value neurons, they interact with this oscillation. So this is the theta oscillation up here, and this is where the neurons show their peak firing rate. So you can see that the neurons are much more likely to show higher firing rates as the oscillation rises compared to when it falls. Now, these are, like I say, these oscillations have been associated with lots of different cognitive processes. One of the problems, though, is that all of this research tends to be very correlative. So what we do is we basically, and there's a problem with that. Behavior is very complex. It's high dimensional. There are a lot of different behavioral measurements that you can take. These oscillations are also very complex. They are also high dimensional, and there's a lot of measurements that you can take. And when you correlate one high dimensional measurement with another high dimensional measurement, your chance of seeing correlations are very high. But it doesn't necessarily mean that those correlations are real, or that they even have any kind of functional significance. What we really want is a causal manipulation of these rhythms. But one problem with that is that our current neuroscientific methods are not precise enough to manipulate a specific neural rhythm without also affecting other underlying rhythms or the underlying neuronal spiking. So things like pharmacological manipulation or optogenetics or electrical stimulation will all affect both the all of the neural rhythms and also the neuronal spiking. So to solve this problem, we developed what we call a closed-loop microstimulation protocol, where we essentially record the theta oscillation in real time, and we use that as a signal to apply our microstimulation. So all of this is taking place in the orbitofrontal cortex. We record the theta rhythm, and every time theta rhythm peaks, we apply a pulse of stimulation. And what that does is it really disrupts the theta rhythm without affecting any other rhythm or the individual neurons. So this is what happens to the animal when we do this. So this is how many trials it takes them to adjust their behavior when the reward contingencies start changing. And you can see that our animals are really good at this task. You know, they adjust their behavior in just a few trials. But when we apply this theta stimulation, they're really, really impaired. They cannot do this very, very simple task. Now, as a control, we also did the exact same manipulation, but this time we used the beta oscillation. So we recorded the beta oscillation and stimulated at the peak of that oscillation. And we have no effect on behavior. And this is a very powerful control because the beta oscillation is 3 times faster than the theta oscillation. And so that means that we're applying 3 times as much current into orbitofrontal cortex. And yet we have no effect on the behavior. And in fact, if we go back and look at if we just do what we call open loop stimulation, which is where we just stimulate at random on the theta oscillation, we can show that only the stimulation pulses that arrive at the peak of the oscillation have this detrimental effect on the animal's choice behavior. Okay, so to summarize these findings, learning about rewards in your environment involves this increase in the theta oscillation in the orbitofrontal cortex, and closed loop stimulation of this oscillation can disrupt reward-based learning. Now, when you start to think about the theta oscillation, we start thinking about the hippocampus. And the reason for that is that the hippocampus has a very strong theta oscillation, and we know that it projects up to the orbitofrontal cortex. So the question was, is the hippocampus involved? And if it is involved, what exactly is it doing? So just to introduce you now to the hippocampus is this old, evolutionary, old structure that's buried down in the temporal lobes. It gets its name from the Greek for seahorse, because the Greeks thought it looked like a seahorse in the brain. And damage to the hippocampus is this is one of the most famous case studies in neuroscience. It involves patient HM. So HM had epilepsy, and at the age of 27, he had his hippocampus removed in order to control that epilepsy. So here's a normal person with their hippocampus intact, and here's HM with his hippocampus missing. And the surgery stopped the seizures, but from that day on, HM had very profound amnesia. Now, what is amnesia like? Everybody knows that it is not being able to form new memories, but what is the experience of having amnesia like? Well, this is an example. This is a patient, Clive Wearing from England. He contracted encephalitis, and that left him very amnesic. This is his journal. I don't know if you can read it, but basically it consists of him writing the time and then saying something like, I am now awake for the first time. And then that time gets crossed out and a new time is written in. And he writes, I am now perfectly awake, first time for years and months. And this is what he spends the entire day doing. And he has notebooks and notebooks and notebooks because he's been amnesic for so many years at this point. So you're going to hear him describing what it's like. Essentially, because he has no past, he has no memory of the past whatsoever. To him, it feels like he's just recovered from being unconscious. And that's what his day-to-day experience consists of. So you'll hear him describe it now.
I've nothing to say about it. It's just like death. No thoughts of any kind, no dreams, no difference between day and night, no sight, no sound, no taste, no touch, no smell, exactly like death. No difference between day and night. No thoughts, nothing. No dreams, nothing at all. Any question you have, its answer, I don't know. There's nothing to say. No dreams, no sight, no sound, no taste, no touch, no smell, nothing at all, no thoughts, nothing.
Since how long ago?
For years, that's all I know. Whole time I've been ill, nothing at all, no thoughts, nothing.
What's it like now?
I can see. First time I had any evidence I was alive.
Do you feel sort of normal now?
Yes. Since I sat down here, I don't remember sitting down now.
You don't remember sitting down?
No, I never see any human being now. I can see you fully open now.
So you can see that his thoughts, his speech is very repetitive. And that's because he doesn't remember what he just told you a couple of minutes ago. And he's kind of locked in this, perpetual present moment. Now, when we look in animals and ask what the hippocampus is doing, we find that it's really important for spatial learning, for knowing whereabouts you are in an environment. And this is a task that has been used in many labs to study spatial learning. It's called the Morris Water Maze. There is a hidden platform in this pool of water, and the animal basically has to swim around to find the location of that platform. And then once he's found that location, he's able to swim on other trials, he can just swim straight to the platform. Now, in animals with hippocampal damage, they just don't learn this task. And every single trial, they have to swim around and search for the hidden platform. And if we look at what hippocampal neurons are doing, they essentially encode a map of the environment, a spatial map. So this is an animal. In this experiment, the animal is running around a square box. And each of these colored squares indicates the activity of a single hippocampal neuron. The color indicates its firing rate. And it shows you how the neuron fires depending on where the animal is in the box. And you can see that each of these neurons fires when the animal is in a specific part of the box. And so taken together, these neurons provide the animal, it's kind of like a GPS system for the brain. It's essentially telling the animal where it is all the time. And this is work that was done by John O'Keefe, and along with Edvard and May-Britt Moser, they got the Nobel Prize in 2014. The Mosers discovered grid cells that are sort of more complex place cells. These are in the area that projects the entorhinal cortex, which projects to the hippocampus. So that's all very interesting, but what does it have to do with our task in which we're manipulating? We're not manipulating space, we're manipulating value. Well, the first thing that we noticed is that when the animals are adjusting their behavior, when they're learning, the hippocampus also shows this theta oscillation. And the theta oscillations in these two structures, they synchronize. So the two structures start firing in unison. And you can see that in this figure here. This is a measure of how synchronized, the color is a measure of how synchronized the two areas are. This is when the animal's performance starts dropping because the reward contingencies have changed. And when the animal detects that the reward contingencies have changed and starts adjusting its behavior, we see this synchronization of the theta oscillation between these two areas. Now, if we do our same stimulation manipulation, if we now go into the hippocampus and do this closed loop theta stimulation, we severely disrupt the animal's ability to learn. So we're disrupting theta in either the orbitofrontal cortex or the hippocampus, prevents them learning about reward. And we can also figure out the direction of this effect. So we can essentially look at the lag, how long it takes one area to influence another area. And when we stimulate in the hippocampus, we disrupt both the hippocampus transmission to the orbitofrontal cortex, but also the orbitofrontal transmission back to the hippocampus. Whereas if we stimulate in the orbitofrontal cortex, we only affect the orbitofrontal to the hippocampus direction. The hippocampus to the orbitofrontal is intact. So what this tells us is what we suspected originally, that the theta oscillation originates in the hippocampus and it's synchronizing, it's providing the orbitofrontal cortex with the same oscillation and even synchronizing that oscillation. But the question is really, what are the neurons doing? Why are the hippocampal neurons important for doing this task? If we look at the firing of hippocampal neurons, and this is their firing across the entire session, we see something quite striking, which is that their firing rates are almost zero for most of the session. And then there are just these random points in the session where the neuron begins to fire very strongly. And so we wanted to know what do those points mean, what's being encoded there. So remember what the task is. We have these three pictures. They're changing in value. And we realized that you can represent this as an abstract value space. So in this abstract value space, each of these pictures, the value represents one axis. So any point in this value space specifies the relative value of the three pictures to one another. And you can now, if we represent this, these changes in this value space, we actually get a circular trajectory. So we actually designed this so that we could move in a circular pattern around this abstract value space. Now, what are the hippocampal neurons doing? Well, when we recorded their activity, we found that they would fire in a specific location within this value space. So this is one neuron. You can see that it fires in this upper right part of the value space. This is the first time the animal went around the trajectory. And then this is the second time the animal went around the trajectory. And you can see that it fires in pretty much the same location on both of those trajectories. And that was also true in this neuron here, but it has a different location. It's firing for this lower right location. And then we have a third neuron, this one firing in the upper left. And in fact, when we recorded from a whole bunch of hippocampal neurons, we found that pretty much every location in space, in this value space, was represented. There was some neurons that would fire in every single one of these locations. So just as the hippocampal space cells are representing the spatial location of an animal when it's performing a task, these value neurons are representing the location in value space. So what does that mean? Well, this was actually an idea that was put forward about 30 years ago now by Howard Eichenbaum. He argued that what was going on in the hippocampus was a relational code. And the reason that you see this, the reason that this relational code looks like a spatial map is because space is inherently relational. We remember the location of things in space via their location relative to other objects. So here's our rat. He's staring at this environment, and he's encoding the relationships between each of the objects that he can see, and that gives him this spatial map. Now, value is also relational. We encode the value of items relative to one another. And so it seems that the hippocampus is also employed for that reason. So the animal can use this relational code for any kind of relational information. Okay, so bringing it all together now, this is what we think is happening when animals learn about rewards or humans learn about rewards. We think that the orbitofrontal cortex is critical for decision making. It essentially enables us to pick one option relative to another option by knowing the values of those options. The hippocampus, though, also provides value information. It basically tells the orbitofrontal cortex the relative value of objects that the animal is experiencing. And they communicate this information to one another by synchronizing the theta oscillation between the two structures. And so what happens when we stimulate is that we push the oscillation in one area out of alignment with the other area. The two areas can no longer communicate with one another, and the animal can no longer make good choices. Now, what does this all mean? What does this all have to do with the original point of the talk, which was, the development of brain-machine interfaces? Well, you can begin to see how we can take all of this information and start to build our brain-machine interface device. So imagine that we have some behavior that we want to modify. In this case, we have a person engaging in drug-taking behavior. What we can do with the device is we can read out their decisions and detect when they're considering making this maladaptive choice. And we can specifically disrupt that choice by targeting the theta oscillation. And in this way, we hope to be able to bias choices away from maladaptive decisions and towards more optimal decisions. So that's what we're currently working on in my lab. One of the issues that we face is that the performance of our decoders that are able to do this depend upon the number of value coding neurons that you can record simultaneously. The more neurons, you can see that in this plot here, the more neurons we record, the better our decoder performs. This was from our old paper where we were only recording from about 20 neurons at a time. These days, though, we use these probes here, which have multiple contacts. Instead of just having a single wire, within that single wire, there are embedded 16 even smaller wires. So you can record from 16 different neurons simultaneously using the same probe. And this has enabled us, here we have 4 of these probes lowered into the brain, and we're recording now typically from 100 to 200 neurons in a single day. This is what we're currently working on, though. This is the Neuro Pixels probe. This is now a silicon probe, and instead of having 16 contacts, it has 384 contacts. And this is some pilot data where we recorded from about 300 neurons simultaneously. And currently in development is the primate pixels probe, which has now 4,000 channels along its length. And we hope that we'll be able to use that to record from thousands of neurons simultaneously, really improve the performance of our decoding, and be able to much more accurately understand how these decisions are unfolding so that we can meaningfully interact with them. So I'll finish up there so that we have time for some questions. I just want to acknowledge everybody in my lab that did this work, particularly Eric Knudsen, who was the postdoc in the lab. He did all of the stimulation studies and also the hippocampal studies that I told you about today. And yeah, I'll be happy to take questions.
Спасибо большое. В чате пишут много о том, что тема действительно актуальна и интересна, а также отмечают, что вдвойне интересно слушать про такие исследования на животных, когда понимаешь, что даже обезьяна умеет управлять роботизированной рукой, то невольно чувствуешь в себе силы для того, чтобы совершать новые свершения самостоятельно. Переходя к вопросам, подскажите, пожалуйста, правильно ли можно понять, что в самом мозге нет рецепторов, которые отвечают за боль, и именно это позволяет накладывать электроды прямо на мозг?
That's right. So all of our pain receptors are in the periphery, right? They're on our skin, or they're in our organs or muscles, and so the brain is processing all of that pain information. So all of that information goes into the brain, and that's where pain is represented. But inside the brain itself, there are no pain receptors. So you know, a lot of those, for instance, the deep brain stimulation electrodes in humans, when you put the electrode into the patient's brain, you don't use any anesthesia, and the patient is wide awake. So there is anesthesia for going through the skull, and there's anesthesia for going through the protective covering of the brain, which is called the dura. But once the electrode is in, you can lower that electrode, and the patient has no feeling whatsoever and doesn't know it's in there.
А если, например, животное сыто, как его можно еще замотивировать проходить эксперимент? На что дикие животные, кроме еды, могут откликаться? И могут ли?
So the animals are we typically use fluid. The animals are actually restricted. They're not allowed to drink for a few hours before the experiment to make sure that they're thirsty. So that's what we use. The reason is basically it's an easy way to motivate an animal without it being physiologically stressful. But other people have used all kinds of rewards. So some people have used, definitely some people have used food. Some people have used access to a sexual partner. Some people have used direct stimulation of the brain. So you can actually get an animal to, if you put an electrode in that will stimulate the animal's reward centers, the animal will actually work in order to earn direct stimulation of its reward centers. And importantly for our work, a lot of our work is focused on addiction. Animals also get addicted to drugs. If you provide animals with drugs, they will take them. They get pleasure from taking drugs. This is true for rodents, it's true for primates. It's true for every mammal that we tested, the people have tested it on. And so it gives us a really good model for human addiction, because there's this overlap, there's this similarity between humans and animals.
То есть потенциально можно сказать, что мы можем бороться с зависимостью человека при помощи вживления электродов на мозг.
Yeah, that's the idea. And so our lab is really focused on sort of demonstrating that is possible, and then hopefully other labs will take that and, you know, try to develop these devices for use in humans.
Это очень интересная и действительно очень прикладная тема. А можно ли с помощью электродов влиять на чувства человека? То есть сделать так, чтобы он получил удовольствие, допустим, такое же, которое получает от еды или алкоголя или психоактивных веществ?
We definitely can. So we know that directly stimulating the reward centers is rewarding to people in the same way that, you know, food and drink is rewarding. In fact, it's even more rewarding. It's like hyper rewarding. I have some colleagues that are working on anxiety and depression. And so what they've been able to do is use stimulating electrodes to adjust people's physiological arousal state. So you essentially record from areas of the brain that we know are responsible for arousal. So this is things like, you know, increasing heart rate, you know, your palms get sweaty, all of that's under the control of the brain. And you can put electrodes in there that are detecting whether your heart rate has gone up and then slow it back down again. And the idea there is to sort of help people with things like panic attacks, where you would be able to, you know, clamp down this maladaptive physiological response.
А знакомы ли вы с экспериментами академика Натальи Бехтеревой?
I don't, sorry.
Еще такой вопрос. На данный момент понятно ли уже ученым, каким образом осуществляется синхронизация между гиппокампусом и орбитофронтальной зоной? То есть как самонастраивающееся радио?
Yeah, that's a really good question. We don't fully understand the mechanisms that generate local field potentials, but one of the very powerful synchronizers in the brain is something called coupled oscillation. So coupled oscillation was originally discovered by Huygens, who was a 17th century physicist, I think. And he was looking at pendulum clocks that were on the wall of his house. And he noticed that those pendulum clocks would begin to synchronize. And we now know that that is this phenomenon called entrainment. And so basically, if a neuron, if anything has an oscillation and it is somehow coupled in a way that can influence another oscillation, those two oscillations will eventually synchronize because they kind of correct one another's oscillation. So if one of them starts going too fast because the other one is going slower, it will pull the other one back into the average of the two oscillations. It's a similar phenomenon to you know, pushing a child on a swing. If you time your push exactly correctly, it has a much bigger influence than if you mistime it. And so the brain is, neurons in the brain are essentially coupled oscillators. If one neuron is firing at a particular firing rate and each neuron is connected to many other neurons, and those firing rates influence whether the other neuron fires or not, eventually they all start firing in rhythm. And that's when you get these oscillations generated. So we know that the hippocampus is strongly connected to the orbitofrontal cortex. So in other words, the neurons in the hippocampus, they send out long projections that interact directly with orbitofrontal neurons. And actually, vice versa as well. The orbitofrontal is sending projections directly to the hippocampus. And so because they're connected with one another, if an oscillation takes place in one area of the brain, it will naturally tend to cause the oscillation to occur in the other area of the brain.
Есть ли на данный момент исследования, которые показывают влияние медитации на активность мозга?
There is. I am not familiar with it myself. I think it has to do with the alpha rhythm. I know that a neuroscientist called Chris Moore at MIT has studied the particular rhythms that are generated in meditation, and in fact, so each year we have a very big neuroscience conference. It's about 40,000 neuroscientists meet. And one year the Dalai Lama came to the conference and spoke. And that was specifically to understand the neural, what's going on in the brain during meditation. So there is there are people researching it, and there are also people communicating with the Buddhist community.
При стимуляции мозга электродами, как при применении психотропных веществ, наносится ли организму такой же вред, как при принятии реальных психотропных веществ?
That's a very good question. So in terms of psychoactive drugs, it depends very much on what kind of psychoactive drug you're taking. The ones that don't seem to do very much harm are things like, I would say nicotine. You know, nicotine has other harmful effects, but not on the brain. Caffeine, marijuana, LSD, any hallucinogen, they're fine. But then there's a whole bunch that are really harmful for the brain: opiates, cocaine, methamphetamine, MDMA. Let me see, and alcohol. So all of those are terrible for the brain. So it depends. In terms of putting electrodes in, the brain isn't harmed, but the brain does generate not an immune response, a sort of an inflammatory response. So what happens is cells called glial cells begin to coat the electrode. Now that doesn't harm the brain, but it means that you can't get very good recordings from the electrode anymore, because it's sort of insulated by all of these cells that have, you know, stuck around it. So that's been a real problem. It is a real problem for the deep brain stimulators. Typically what we find is that they work very well when they're first in the brain, and then over the years that the patient has them, you constantly have to turn up the current because of the cells that are built up around the electrode. Currently, the way that we're trying to tackle that is to make the electrodes very, very small indeed, so that there's evidence that if you get down to the nanoscale, the brain, which is possible, you can do recordings with nanoscale size electronics. The brain no longer recognizes them as a foreign body. It's too small for the inflammatory response to recognize as a foreign body. And so that's one possibility for developing electrodes that you could leave in the brain with no harmful consequences whatsoever.
Исходя из тех психоактивных веществ, которые вы перечислили, правильно ли получается, что алкоголь наносит мозгу больше вред, чем, например, марихуана?
Yeah, I think that's definitely true. You know, we see neuronal loss with alcohol. We see changes in, and you know, this is in alcoholics, right? So this is very, very strong alcohol use. But the other big problem, the other big health consequence of alcohol is, you know, children born to alcoholics, so fetal alcohol syndrome. The brains of children with fetal alcohol syndrome are quite seriously impacted. So yeah, the health consequences of alcohol, excessive alcohol consumption, are really quite profound.
Ушиб тяжелой степени лобно-височной доли с комой в несколько дней является повреждением орбитофронтальной коры или нет?
Yeah, so the typical reasons that people have damage to the OFC, to the orbitofrontal cortex, are tumors, strokes. The middle cerebral artery goes up into the comes up into the frontal lobe at the base where the orbitofrontal cortex is. So if you have a stroke there, you'll damage the orbitofrontal cortex. And traumatic brain injury. So if you look at the skull, if you look at the inside of the cranium, most of the cranium is smooth. And the exception to that is where the orbitofrontal cortex is. It rests on top of the eye orbits and on top of the nasal cavity. And all of that has its very sharp, bony ridges. And so even if you have a head injury that say hits the back of your head, the damage is often to the orbitofrontal cortex because the brain shoots forward and it gets and it hits these jagged edges. In fact, the person who showed this was I think a 19th century physician. And what he did is he basically took human skulls and filled them with jello. He blocked off all the exits from the skull and then filled them with jello and shook them and then broke it open to see where the damage had happened. And it was almost always the orbitofrontal cortex. So certainly we see a lot of these deficits, we see a lot of these decision-making problems in patients that have suffered traumatic brain injury.
Получается, что нельзя бороться с зависимостью, просто давая нужный импульс мозгу через электроды, так как организм вырабатывает некий иммунитет.
Well, I think this is, if you think about what you're trying to do with electrodes, you're trying to meaningfully interact with the ongoing patterns of activity in the brain. So it's almost like we're learning to program a computer. And once you learn the programming language, what is available to you is essentially unlimited. So we're working on this for what we want to do, but other people are working on it for other issues. And we hope to be, a lot of those problems that we currently face are solvable problems. So for instance, one of the big problems with putting electrodes in the brain is that it's a route for infection to get into the brain. Obviously that's bad, but people are working on wireless recordings and wireless recording technology. And another problem is the buildup of tissue on the electrodes, but people are working on miniaturizing everything so that that buildup doesn't take place. So these are not insurmountable problems. We haven't surmounted them yet, but and I'm sure that, you know, as we solve one set of problems, we'll encounter other problems. But the potential of this is what we're all excited about. It's a much more precise way of interacting with the brain compared to, say, neuropsychiatric medication.
Спасибо большое за такую интересную встречу. Спасибо большое за развернутые ответы на вопросы. На этом наше время, к сожалению, заканчивается. Спасибо большое за взгляд с научной стороны, для того, чтобы посмотреть, что на самом деле происходит внутри. Это действительно очень интересно, и, конечно, в том числе для психолога это очень важные компетенции, которые могут показать не только внешнее наблюдаемое поведение, но и внутреннее поведение, те процессы, которые происходят внутри, и что, конечно, тоже стоит учитывать, когда мы говорим о такой непростой теме. Спасибо большое за встречу, спасибо большое участникам за вопросы. На этом наше время заканчивается. Следующий вторник, в то же самое время по Москве, мы с вами встретимся в рамках темы, посвященной профилактике подростковой депрессии. Тема также является прикладной, поэтому обязательно подключайтесь. Вторник, 19:00 по Москве. Еще раз огромное спасибо за участие, спасибо большое за такую интересную презентацию и всего хорошего.